So Mike Vecchione says what you're looking for is called a dog on a watertight door.
What? Oh, yes. Thank you, sir. My shirt got caught on the dog on the watertight door, and it ripped.
I see. So I believe our transect we've got something down below us.
Maybe some...
Watch leads inshore. Just would like to let you know, 1433 UTC, we will officially start the 300-meter transect 1.
Thank you.
Yeah, I would love that. Yeah.
Transecting.
Thank you.
Copy. Pilot is on OKEX. I'll try to remember to turn it off when I'm done transecting.
Thanks, pilot.
Yeah.
We'll remind you if you get, you know.
Copy.
So, yeah, I don't see much right now. It's just marine snow.
Looks like a lot of marine snow.
You can still see the light from the surface, I think.
Mm-hmm.
Either that or it's just I'm just seeing Sirius coming down. I don't know if our cameras would pick up too much light at this depth.
Something crustacean moving. Yeah. As I said, this depth can be a bit challenging to find things. I think there's something coming up there.
There's something at noon.
Siphonophore.
Chain here.
I think it's just schmutz.
Yeah.
Oh, yeah. Just marine snow, snot and whatnot.
Sea snot.
Mucus on its way to the seafloor.
Yeah, lighting looks fine. Video come in.
Oh, there's something there. We got a jelly dead center, maybe? Or a dead larvation house? What do we
No,
got?
I think what we
jelly?
have is a comb jelly. That looks like Thalassa calyce. That's an unusual lobate jelly that actually takes on the form of a... This really does look a lot more like a medusa than comb jellies typically do, but...
Come and push out.
And it has basically kind of a bell. That's what makes it look Medusa-like.
Adjust.
And this, it closes up that bell around prey and then waits for the prey to bounce on off of its stomachs, off of its lips, and then eats it.
Siphonophore?
Yeah, we have a siphonophore there. I think that's probably Nanomia. Really.
Come out.
That's in the group Physinectae.
No, just stay there.
Which has a float and the swimming bills.
Okay.
So, if any of you saw the little benthic siphonophore, the dandelion siphonophore that we've seen a couple times on our bottom dives, those are in that same group. It has a big fat float on the top and then a bunch of swimming bells.
Little bits running by. Maybe some, I don't know what they'd be that small.
Amphipods or copepods, most likely.
Dot.
There you guys
Little
go.
white dots.
Good eyes.
You can see maybe copepods in the foreground. I don't know what that is. Looks teeny tiny. Oh my goodness.
Trying to get a beat.
Boreal.
Oh, it moved.
Maybe.
It's part of the game here.
Yep.
Yep.
There's something.
Yeah,
Center
video.
screen.
Take it.
Schmutz.
Schmutz.
Come on.
Amorphous blobs of mucus.
That's right. Transferring carbon and nutrients to the seafloor. Mike Vecchione says what we were focusing in on might have been a larval fish.
Take this.
Oh, cool. Oh, here's something.
Oh,
Maybe an egg? What is that?
oh, I think that might have been a foram
Oh,
or
cool.
Take it.
or radiolarian. Looks like a single-celled organism. Yep,
It's very small.
It's very tiny.
very small.
Yeah. You take it, let it go. I gotta turn back to port. Coming back around. Pushing ahead, Fernando. Okay. Use the
You
flares.
got something here, siphonophore there.
Looks like it's running away.
Okay. Yeah, it ran away. Out of my light pool. I'll keep my camera in my light pool here.
There's something at yep.
I think that's
Ooh.
Where'd it go?
Nenomia again.
It's
Little-
diving on me.
Siphonophore.
Small siphonophore, yeah.
Yeah. Oh, that one was moving.
If I was right, and it is Nenomia, that is one of the most abundant organisms in the shallow
Something at
mesopelagic.
9, something long at 9.
Ooh.
Yeah, it's too close.
Yeah, I don't see it. Yeah, not sure what the flare is from, Ron. Might be the start of a craft job. I come in.
Yep, center screen.
So
There's another one of those.
Yep.
Yeah, I'm glad we're getting another look at that. You know, this could be a Phaedarian.
What's that?
It's a single-celled organism, and they basically have a bit of
Thanks, Ron. Sorry.
waste product that they have separate from them. So inside the cell, did you see there was sort of a central area, and then there was a little dot just adjacent to that?
Yeah.
That's the telltale signs of what a Phaedarian.
Okay. So single-celled plankton.
Yeah, single-cell plankton with pseudopods.
Larger increase the
Used
ship
to be
speed.
associated with
Decimal one more. So you're
radiolarians, but the sort of molecular genetics has suggested that it's a separate group.
We got maybe a copepod coming in. Oh, that's out. Let's see.
okay.
Yep. Siphonophore schmutz.
I think it's a schmutz that looks like a siphonophore, right? Just a long.
Bye.
We're having the ship speed up to slow us down a little bit.
Okay. Oh, you got something that, yep.
Trying here.
Yep, schmutz.
It's
Radio. What is that, a larvation house?
could be a larvation house, yeah, but it
Let it go.
doesn't seem quite organized the way I would expect. But
Schmutz. It's schmutz.
A lot of
And it's schmutz.
A lot of schmutz at 300 meters today. I gotta push out a little bit, Ron.
There's one right there.
And the brakes.
ooh,
What
that
is
looks
that?
like a medusa.
Wait for it. Wait for it. I think it's close. Do you agree, Ron?
Schmutz.
Oh, good try.
Okay, I'm gonna push out, Roland. Find me something amazing, Roland.
The amazing is out there.
Oh, I just saw something cool, but it went off screen. It had flashy lights. Reflective.
Ctenophore refractions.
Yeah, probably.
Yeah. It's more flares, though. Kind of right in the middle of the screen is what I'm looking for. Otherwise, it floats up or it floats down.
Saw another small siphonophore going off to the right.
Oh, yeah, there's something up top,
above us.
too.
That
Oh.
11 o'clock?
Yeah.
That
Video.
looks like a little ctenophore. Yep, we have a comb jelly.
Nice.
Take more if you got it. Let me know if I'm getting closer or further. I don't have a grasp on distance here.
This is really nice.
Cool.
Understood.
It's a lobate ctenophore. I think this is, this sort of looks like Oceropsis or Bolenopsis. I'm not very good with these lobates, but this is good enough for us to definitely identify. You can see it cruising through the water there, sort of,
Okay.
Encountering
Down a
food.
little bit, Fernando.
And it's
We're
going.
looking right down on it.
Is it going mouth first through the water?
It's going mouth first through the water column, and its lobes are sort of held up above and below from our view, and the water's going into the mouth region, which is at the end of the lobes there, close to where you can sort of see the gut central.
So upper part.
Yeah, that was a great view of that.
Okay.
And
We got to reset.
sit there and think about that.
Yeah. Copy, Fernando.
Oh, I saw
Yeah,
Medusa.
I saw the jelly too. I wish I had time. I'm resetting right now. The ship's moving, so now the water will slow down,
Okay.
which is good. We'll have more time. Okay, here we go. I'm reset now.
Oh.
Big self
Yep,
chain.
yep.
You can go for the head right there. Should be the ball.
Yeah.
There we go. That's neat.
Oh, that is nice. So this is another siphonophore. And I'm trying
Cool. Whoop.
It looks like Hippopodius, maybe. Hippopodius hippopus. I'm not sure, though. Were those pulsing?
I saw one, yeah.
Mm-hmm.
Resetting.
It looked like they were pulsing, and maybe just a couple of them.
Mm-hmm.
But there was no float, right? Yeah.
It looked like no, there was no big ball float like they have on the bottom ones. It looked like it was just the bell, and part of that bell was pulsating.
So that's how we tell apart a group that we call Chalicophora.
That's what a Cadignath.
Our first Cadignath. So these.
That's very small.
Arrowworm.
Arrowworm, exactly.
It's crazy how sometimes they just disappear.
So abundant, and yet we only know about 130 species of them.
How many do they think there are?
I'm not sure how much people have estimate. We still need to know about their diversity.
Ooh, what's that?
Ooh, let's.
Oh, schmutz.
It's like maybe an old larvacean house.
Chain of dots.
Chain of dots, yeah. Ooh. Oh, cool. Looks like a leftover. Oh, look how neat. It's moving.
It is definitely moving. I think we may.
What is going on here?
Understood.
What is that?
I think
I mean, it's got to be a siphonophore.
I
some
think those are
kind.
parts of a siphonophore, that have budded off.
It's going down.
Sweet.
And rotating down to keep it in view.
It has a little bit of movement, like,
Yeah, coordinated.
Mm-hmm.
Huh. Well, Mike thinks that might be a salp chain, but it wasn't looking like a salp chain to me, except
Good reset.
the way it was pulsing. But maybe it was a salp chain. Salp we would recognize by having a hole at the front and the back.
Okay.
Copy. 45.
And a salp is a tunicate.
As a tunicate, exactly. So
It's
it's actually a relative of the Larvaceans.
Yep. and the chordata, which has in its
Has a notochord in
Can dig
its
a little bit.
exactly.
in its larval stage.
Yes, except for the Larvaceans, who they retain that to their adult stage. But
Your phylogenetically oldest living relative,
that's right, our closest
I see it too.
closest living relative.
invert.
Potential jelly.
Oh yeah.
Yeah, that's a nice one. That is the Narcomedusa
closer, aren't I?
solmissus. Pretty common site. I think we saw one yesterday during the beginning of yesterday's dive as well. And
I want to push towards it a
This
little bit. Try and get more light on it.
whole group Narcomedusae are
Oh.
readily identifiable
Okay.
by their tentacles being on the tops of their bells, exiting the tops of their bells. And
Okay.
those are
It's worth a try.
more cruisers. Those tend to go through the water with their tentacles spread out in front of them, looking to encounter potential prey.
Schmutz.
Yeah.
Schmutz.
Yeah. Do a compilation of Schmutz.
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 It's all schmutz.
Yeah, we get into the deeper, darker waters. We won't be saying it as much because
Yeah.
we'll find other things to look at.
Feels better.
Ooh, there's something.
Huh.
Focus. That was hard to
A
focus
long
on.
way away. Or small.
I think it was too close.
Yeah, that one was too close.
It's just out of our focusing range.
Yeah, I got in the lens sometimes. Yeah, I see that too.
There's a little white thing. Little bug. What do you got? What
Oh,
are
is this-
you?
Oh, that's
Oh, cool.
okay.
Look how neat. What is that?
I'm a little bit stumped. The two trailing tentacles. You can see the
Like a mucus net?
mucus
sitting
net.
above its
You
head?
know,
It's a
some
pteropod.
pteropods have
Pteropod.
mucus nets. This is so this is a thecosome, right, Mike?
Yeah, it's a thecosome. It's a cavilliniid pteropod.
That's great, and you can see that this. I haven't seen that too
They
often.
do feed with mucus nets, but
Not a little bit.
much of what you're seeing is it extrudes its body out of the shell, and when it's got the body out of the shell,
Okay, let it go.
the whole shell sort of gets wrapped up in the body.
Thanks, video.
Oh, that was really nice.
What are the two tentacles? Are they actually tentacles coming out the bottom?
Okay.
The two long
Well,
hairs?
they used to be called balancers, but they extend from the pointed corners of the shell.
Oh, neat.
And it's not clear what they're for, but they use them when they're floating motionless like that. So they might have something to do with buoyancy.
And these are feeding on phytoplankton mostly.
Well, they're feeding on microscopic stuff. So
Okay.
little phytoplankton and also larval eukaryotes and just whatever tiny stuff gets tangled up in the mucus.
That was really cool.
Yeah, it was a nice sighting.
3 o'clock, white thing.
Copy. Video.
There's a little guy. Something. Cool.
Okay.
That's
let's get
teeny
it
tiny.
He's very small.
I wonder if... Is that another pteropod?
Oh,
Oh,
bye.
Yeah, that looks like
that
another pteropod. That
Cuvirini?
looks like a cigar pteropod.
can you say that again?
Cuviorina.
Cuviorina, yeah, that's what I was...
I think it looks like Cuviorina. It's also, now it's decided to sink, and it's sinking like a pteropod as well.
So we saw a lot
It's
of...
called the cigar pteropod.
The cigar pteropod, yeah. We saw a lot of their shells among the pteropod hash,
You're right, the real long ones.
the long, skinny
Yeah,
ones that
cool.
was look like a little tube.
That's nice to see what they look like, alive. That's pretty cool.
Exactly.
Yeah, there are other long, skinny ones that look like tubes as well.
Oh,
Let's-
good to know.
Oh, no, that's a ctenophore. I see it flashing.
Yep, Tina Ford, bottom right.
Cool. He's different.
Was that wrong?
It's another lobate Tina Ford.
I'll let it go.
So, all right. So we're pushing ahead. Just joining us, this is our 300-meter transect through the water column. We're going to be doing 300 meters, 500 meters, 700 meters, 900 meters, and then about 10 meters off the bottom floor today. And that's going to be, the bottom is at about 1,000 meters. See something moving dead ahead.
Come in.
It's swimming upwards. It's a little high.
I don't see a video.
That's all right. Let's go see what that is.
There's two, actually. There's one below this.
Okay, I believe we're looking at there. It's not moving at all. That's going to be a colonial radiolarian. It's normally a single-celled organism, but they can make these little groups.
I'm working on it. Yeah.
There we see another Solmissus. That's another Narcomedusa.
It's going to hit my prop wash here. We'll let it go.
And the interesting things about Narcomedusae came out of a big study of feeding interactions they'd observed in Monterey Bay Canyon. Basically showed that Narcomedusae have the biggest, sort of like the most complex food webs that were documented. The most number of different things that they eat and that eat them. So really critical players in this dynamic water column food web. Still
Let's go
hunting.
for the
Oh,
fish.
there we have a nice little fish.
Take more.
Oh, no. That's a Okeanos doing its best fish impression for us. This must be a pretty nice big guy. You can see it's going at the tail end is to the right, and that's where its little gonads are showing bright white, and its head is at the top left end there.
Let it go. That's pretty big. Okay. Moving on.
Or schmutz.
Yep.
Another Okeanos went by the top there.
Okay.
That's schmutz again. Ooh, just went by the bottom, missed something.
What's that?
Maybe a Medusa. It's marine snow.
Shoot.
Yep, more.
Oh, I have a feeling about this one. Come in.
Good job, good job. So here we have a really nice view of this, again, Narcomedusa. Trying to figure out what's going on with a couple of its tentacles. Looked a little out of place or something.
Another one. Go for it. Try
There
to
we go.
Try to get more light on it. I don't know how close I am to him. Oh, okay, sorry. So transparent.
It is so
Backing out.
transparent. Does not want to be seen.
Turn to port.
About
Evasive
Nice.
maneuvers. Okay. Thanks, video. Go back to starboard, Fernando.
with
Okay. Pushing out. Oh, little baby jelly.
small Medusa.
Okay.
If you're just tuning into this NOAA Ocean Exploration transect through the water column, we will be going to deeper depths, and at each depth it becomes a little bit easier to see the organisms. This first 300 meter depth, there's still some light coming from above, and it's makes it more difficult to get the organisms to pop out and see very clearly.
You on it?
But we're still seeing some interesting things here. What do we got? It's nice. This is a siphonophore. You can see it's got its its stem and all of its zooids were all spread out for hunting, and now it's a runoff. really remarkably maneuverable organisms.
Yeah, I think it's shrubs. Keep going.
Whee, there goes. Another Keatingnath, I think.
We'll let that one go. Oh, come in.
I think we've got another siphonophore there. This one looks a little bit different up front. Someone won bingo.
If it was 064.
Thank you for that. And that's a Cali Coffrin. So that's a siphonophore we hadn't seen yet.
Okay. All right. Are those in DBs? Roland? Never mind. I'm waiting for something amazing. It's gonna pop out.
We're all waiting for the.
Can you give him some more for now? Looks like upper's got a little bit left. Sure. Chris is going to try to take care of that flare for us. 780 RPM.
What's going by?
I think that's the one he's trying to. Yeah. How about the white dot?
Let's take the. That's a little.
Let it go, video.
Yep.
Yeah.
More.
Ooh, what's that little white guy?
Let's take a look.
Little tail. Doing a little.
Now, this looks like it might be a larval fish. Ooh, nice. Could take some good footage of this.
Got maxed out. Yeah.
It drifted out, but it was brief, but I think that will be nice. Someone will have a hypothesis on what that fish is going to grow up to be. Zing. Oh, that's nice.
Take a bell.
Whoa, I was having a little trouble with that one. Could see the cilia moving, but I didn't see them connected up in lines like you would expect in a ctenophore, so I'm curious.
You got something moving there. And it's gone. Wow, that was fast.
Sometimes they just disappear. Come in on that weight. I think it's moving. Not sure.
You missed a nice little fish larva.
Oh, nice. Oh, I'm seeing it in the replay.
Oh.
Oh, what's that?
Yeah, that's a siphonophore. That's the front end of a siphonophore. It's so strange seeing it sit there so quietly. This is one of the diphiids.
Copy.
A really nice view of it. These are sort of rocket ship shaped and usually very fast. You can see its stem is tucked up where the white part's there. That's the part that will come out and dangle out behind the organism with the different zooids that are specialized for feeding or catching food or reproduction. That's really incredible.
We'll let it go video. Thanks.
Okay. And that is definitely good enough for someone to identify a siphonophore person.
Here's something. Thank you.
Thank you.
Oh.
We'll reset, Roland. Okay.
There's some little white things. Maybe there's something.
I'll find both of the white ones until we decide.
That one's
schmutz.
Yeah, we'll let it go.
Double schmutz.
All this organic material.
Mm-hmm.
Yeah, I was moving forward at that time. I'm now drifting with the current, so I should have more time now. What's this down here?
Schmutz.
No,
there's
it's something
a little
swimming.
Yeah, there's
I
a
think
little
that's what they're
shrimp
looking.
or something. There you go, that guy. Copepod. Who's very tiny.
Yep, definitely a copepod.
Cool.
Just cruising around.
Yep, copepods are super abundant and diverse, both.
What's this, Roland?
Many of them are
Siphonophore.
vertical migrators and go up and get the plankton.
Ooh, that's
Yeah.
cool.
That's
He looks different. He's got his comb rows are really weird.
his comb rows are really weird.
A disco ball.
He looks like he's flapping around the side as well. Yeah.
We'll let go video.
Sweet.
Thanks for that.
Any
I'm a little
idea?
bit stumped. No, besides it's ctenophore, I'm pretty sure it was a ctenophore, but and I think it has its lobes out below it.
Push out. Reset. Yeah.
Yeah, I'm gonna have to send that video to somebody.
That's neat. Got a white string.
Oh, we have Nick Bazzaio's in the chat room. He's been working on ctenophores. He's going to get his PhD studying ctenophores.
9
There's
o'clock, a Medusa.
Sol Missus there. Yeah. He said that sort of looked like Diopia. Which
The
is,
disco ball?
Yeah. I can see that. And I definitely trust Nick's eye.
All right, got something here at noon. Little white thing, probably a siphonophore.
Up above, yeah.
There you go. It was just moving. So it looks like schmutz, but it was moving. So.
So it's probably fully deployed there. There it is. Yes, and now I think we've...
Boop, 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 boop.
This is a calicopheran. Oh, nice.
Let it go.
Like a praed. Just with the two big floppy bells up front.
You will be reviewing all these words with me again because I am not spelling anything properly.
That's our job.
Something white.
We work together.
Oh, that's neat. Another one of those siphonophores?
Yeah, another siphon.
Yeah, siphonophore.
Siphonophore for sure. Maybe let's see what's in its tentacles. We might be able to
It's like a shrimp. Catch it. A little shrimp.
I think it's got food. I think we saw.
Had a really long rostrum, it looked like.
Yeah. So that's a nice observation. Is that praed having caught a shrimp in its tentacles? I got a fish, maybe? Or no, it's a Keatingnath.
That's cute.
Yeah.
You got that. We'll keep going.
Good call, pilot.
Oh, yeah. Here we go.
There's something. That's pretty beat up, though.
Yep.
Looks like some tentacles. Schmutz with tentacles.
There may have been some organization there. Sometimes it's hard to make out.
Ooh, ooh, ooh. Something moving. Little fish. Bristlemouth.
I see it.
Oh,
we
it's
lost
foreground,
it.
foreground. Here you go.
Maybe a larval fish, maybe, I think. Maybe another Keatingnath.
It was swimming kind of like, I thought it was bristlemouth because of that weird swimming it's got.
Yeah, it's sort of floppy swimming, right?
Yeah.
Not very graceful in the water.
No, he's not very good at it. I guess they've managed to be the most populous species out here, so.
They obviously
be pretty
swim
fun.
well enough,
Yeah.
exactly.
It works for them.
Those, if we do get a good look at the bristlemouth,
Yeah, the craft looks a lot better now. Craft was blaring.
have a very diverse diet, so
Should
they
help.
can really eat a lot of different things.
There's not much out here, so you really can't be a picky eater.
It's that, yeah.
And it's schmutz. Barbation house, maybe?
Maybe an old
We'll let it go.
Abandoned.
abandoned house.
I agree.
one of the most effective means of transferring carbon down is through those larvacean houses. Larvaceans are really efficient feeders because they are beating and then they really catch a lot of food.
In their filter?
Yeah, in their filter. And some of them build and they abandon the filters really fast, like within hours.
Oh, neat. Something shiny, probably Cadignath. There's a white thing at the bottom.
Roland, come in.
Fuzzy white. And schmutz. What is that?
Yeah.
I don't know.
Yeah,
It's
that
very
could
organized.
be like that might have been one of those radiolarians, Phaedarian type things.
Okay.
There's some trailing, stringy stuff.
So you can-
Abandoned
Just
snot.
when you think, for sure that must be a siphonophore.
Nope.
There's
But
a lot
no.
of abandoned houses here. Copy that.
So I think we got about, saw a fish go by in the bottom left. Cadignath.
I like the little copepods. They kind of like swim and then stop.
Yeah.
They're pretty neat. Like, oh, I'm just a piece of snow. Don't mind me.
Don't mind me. And those copepods are a big part of the vertical migration, diel vertical migration, going up to feed on
Poo.
phytoplankton at night
And poo.
and stuff,
Poo. They eat poo.
and then swimming back down during the day
Yeah.
to
Carpophagic.
deeper days.
They're the ones who eat the poop.
Coprophagic.
Yeah.
It's always a good word.
Cadignath.
Cadignath.
Nath. Copy.
Even the Solmissus has abandoned us for a while. But as I mentioned a couple times, I think it's worth mentioning again, these transects should get progressively more interesting, easier to see, and then also the next transect's going to be in the deep scattering layer, so that should have a higher diversity and abundance of organisms.
And 10 feet above the bottom, 10 meters above the bottom was pretty cool last time.
No, it was really cool. Watch, there'll be nothing there this time.
I know. You never know. Never know. Another Cadignath, maybe?
Yeah. So we have to remember that some of the nice squid we've seen have been just above the bottom,
That's
too.
right.
Including that magna pinna went by, just over that ridge. Ooh. Oh, there we got it. Tina 4, I think.
Take it.
No, I think it's one of those X chromosome looking things,
Oh. Just
which
reflecting.
I don't know their name of, but that's what they look like to me.
Well,
Ooh, what's that?
never got a good look at it.
A little ring of something. Oh.
A ring of schmutz.
Schmutz ring! There's something over there.
Okay, we have about 15 minutes left on this transect.
Thanks, Chris.
Got it.
Sure.
Copy, 1-5.
There's a siphonophore rumbling around down there. There's a medusa or something.
Down at the bottom.
Mm-hmm.
Yeah. I think we
I need to
probably
stop.
missed it. Yeah.
I need to stop looking off to the side because they're hard to zoom in on when they're that, far
Right.
out of view. It's like center screen or nothing.
Pretty much.
Katignath.
Yep. Another key. I think Keating-Nass may take the most seen at this depth.
Mm-hmm. Siphonophore at 6.
Yeah, so I got the chain of dots.
Yep. There you go.
That's that same, I think it's a Praed. Yeah. So we have seen quite a few siphonophores as well. But
Some flashy stuff. Flash, flash.
flash, flash. Bounce the lights back at us. Zzz. Little copepod or something went top to
Swung
bottom
by.
in front of us.
That's weird.
Is that schmutz?
A siphonophore tentacle or something?
I
Abandoned tentacle?
do not know. There
There's
we
another
go.
one.
I think that's that same, that looks like that same Praed. You can get it. Be nice to get a better look at the front end there, the floppy end, if it behaves.
Copy.
Thanks.
Yeah, come out with it.
Hunting, hunting, hunting.
For smushy, clear critters. Not the easiest prey to find.
No, they don't want to be found.
Yeah.
Go for
Ooh,
jelly.
there we go.
There we go. We have a nice little, another Solmissus.
Take one if you got it.
They're Hydra Medusa.
They are in the class Hydrozoa, so that makes them Hydra Medusa. And then there are two big sides to the Hydrozoa. There's a Tracheline group, and that's where this animal falls in. That is all the Narcomedusae and the Trachemedusae, and a really neat group called Limnomedusae. If you've ever seen the flower hat jelly, it's in that group.
I lost that wrong.
And then the other side has most of the organisms that make hydroids. The athecate hydroids, the thecate hydroids, and the siphonophores are on the other side. That might be Thalassocalus-y.
Some little jelly. or a ctenophore. I thought I saw
I think
combs.
that's
but-
combs. Yeah, that's the Medusa-shaped comb jelly, Thalassocalisi. For some reason, every time I go to spell Thalassocalisi, I spell it right, but I
Oh, wow.
don't think I know how it's spelled, but it comes out right. I look at it and say, huh,
You might be right.
that's exactly the way it's supposed to look. But there's a disjunct between the name, Thalassocalisi, and the letters. It's that same siphonophore again.
It's
Wow.
pretty still.
Ooh, yeah, he's nice and still.
So his bells there look like the bells that we saw earlier.
Yeah.
The little
Yes,
ones on a string, but
yep.
we were missing the front end, it looked
And I
like.
think, yeah, this is a Preya, maybe Preya. No, but that's definitely the best look we've gotten at the front swimming ends. Yeah, so I agree. What we saw loose before was not a little salt chain. It was bits of this Preya.
Next video. Coming around.
1028. Got a pretty good look at its morphology. Take another schmutz string of mucus.
Ooh, video, what's this?
I think that might be it. A larvation house or abandoned larvation house. That's right. So we have about 8 minutes left in this transect.
Nice.
It's kind of nice that the ship is flat today, staring at this slowly moving marine snow. Would be harder if we were bouncing around.
Yeah, right. That's pretty smooth today.
Yeah, we got the good weather for the...
Second to last dive.
I think tomorrow too. Tomorrow we'll get back to corals. And on the Million Mounds.
Yeah, hopefully.
More string-like schmutz in the water. Something took off there on that top right. Should be focusing in the center though.
I'm trying not to look. Right? I need like a cover for the corners. Focus in on that like very center.
We call that the science blinders.
The science blinders? Yeah.
Yeah.
I need science blinders.
Anything outside of that is just smuts.
Yeah. No
I don't
data.
know. I think that's where the giant squid's going to be.
Of course.
Of course, I won't see a giant squid now that I said it out loud.
Yeah. Shh.
That would be cool.
Architeuthis.
Yeah, that'd be pretty.
I gave a friend of mine, Edie Witters, gave her the link to sign up.
Mm-hmm.
I hope she's watching today. If you're watching, Edie Witter, hi, Edie.
Ah.
She was one of the people who filmed Architeuthis in situ using the Triton submersibles. And I think that was on National Geographic, so that
There we have
footage
a nice
is out there.
foram.
Sweet.
Haven't seen a lot of them yet.
Foraminifera.
Foraminifera has a central test, like a little shell, and then it has all the, what we can see are all the pseudopods that are extended, the feeding pseudopods.
Bar. Come out. Thanks. Resetting.
And the shells of all these organisms that have them, big and small.
Oh, Ctenophore up top. Sorry.
Oh, yes, this is Velamen or cestum. This is a ribbon-shaped. Nice.
All right, repeat that word. Velamen
Velamen or cestum.
or
Yes.
cestum.
And probably Nick Bizaya will be telling us any second now which of those two it is. And some of these can be really huge. I think the cestum is the big one.
If he survives.
And so they're really highly, so we saw lobate ctenophores
Yeah.
earlier. These are just like super lobate.
Yeah, they're so low bait. They look flat, like a flatworm almost.
Yep. The common name is the Venus girdle. That's right. Nick says it's cestum.
Next video.
Thank you very much, Nick.
3 minutes left in this transect.
All right.
Be advised. Use
Thank
it
you.
wisely.
Copy.
Well, you just did. We got something brand new right there. That was nice.
Ooh, another foreham off the side.
Well, you broke your rule. Started
I
looking to
know.
the side.
I cheated.
This one?
Take a peek.
Little tiny guy.
Take away schmutz.
It's just schmutz.
Go for it.
Oh.
Here's a larvation, yeah? Or is it siphonophore?
I'm wondering if we've got... I think it's a siphonophore.
Yeah,
Yeah.
because of the trail of
And we
tentacles.
saw the whole front, the house there
Was moving a little bit?
was moving. Yeah. So I think that's that same preyed again.
Okay.
Okay.
Cadignath.
Yeah, so Mike Vecchione in the chat is commenting that although we're much deeper than scuba depth, that we're seeing a lot of the same things that the scuba divers see, especially at night when some of these organisms are even higher up in the water column.
Okay.
Okay, that's the end of the 300-meter transect.
And transect. Thank you very much. And
All
then,
right, so where are we going next? 5? Where's that deep scattering line?
yeah, we're going to go to 500. And it's almost perfectly with the deep scattering layer. So that's going to be terrific. And I think it's 10, 15 minutes to drop down and get set up. Be back then.
Yeah, so if you turn on feed 3, you can see our quad screen. On the upper right-hand corner square, it shows is that sonar going through the water column? It's down into the bottom. It shows a sonar from the surface to the bottom. And you can see there's, in the second one over, you can see a little better where Roland is using his mouse. Right about there is about the 500-meter line, and that bright blue is the deep scattering layer. So we are going to take a look at the deep scattering layer. There should be a little more fauna in that zone. That's the area that migrates from during the day and at night. So the diurnal migration, you'll see that move up and down throughout the day. So we're going to go to the 500-meter line and take a look at that next. So we are going to be dropping. We're now at 302 meters. We're going to go deeper. Our ROV pilots need to set up for that. Then we'll take a pre-transect eDNA water sample, at which point we will then do another hour at 500 meters. Okay, so did you say that out loud into the thing or to me? Oh, okay. Yeah, go ahead.
And on stream 3, I took that, what we call the EK60, and put it full screen on that left area where they currently have the mouse at 302 meters. That would be the ROV being reflected by the sonar. And the one right above that at 288 would be the Sirius being reflected on the sonar.
So you can see where we are in the water column. Thanks, Roland.
Sure. And like she said, this is our area of interest that we're headed to next at about 500 meters.
Yep.
We hope to see lots of larger
Critters. Clear critters, not so clear critters.
So as we get close to our next transect depth at 500 meters, we can rehash a little bit about what we saw at 300 meters.
Yeah, sure.
I like the fact that we saw two different kinds of pteropods. One of them set up with its mucus net and some of its body outside the shell, and you could see its little trailing, potentially balancers.
Yeah, that like a long stick that the tightrope walkers hold.
Yeah, I think that was pretty cool. And then seeing cestum, that really odd long ribbon-shaped ctenophore at the end, that was also really nice.
Cool. There's I think probably mostly cadignaths and siphonophores.
Cadignaths and siphonophores for
It was
sure.
the same as the last dive too, where they were the most common. I mean, obviously we didn't sit here and count them all, but
Our impressions were that they were the most abundant organisms that we, at least that are detectable to our eyes.
yeah.
And one calicoferen siphonophore, and then some little nanomia guys. Seem to be more abundant at the beginning of the transect than at the end. And then throughout we saw the Narcomedusa solmissus. Relatively few tentacles, like 10,
Yeah.
12 tentacles.
Pretty separated. A couple the siphonophores with the bells, I like them. With their little flopping bells. They're neat to watch. Fascinating to me.
Absolutely, and we got great footage of a lobate ctenophore that I think was Oceropsis, and I don't know if Nick was with us. Nick Bazzaio was with us at that time, but hopefully I'll make sure he gets a good look at that footage. And we also saw Thalassocalaceae a couple times, 2 or 3 times. That's the lobate. No, sort of
Ctenophore?
another hyper-lobate ctenophore. That's
Yeah. Oh, the real long
the
one.
Medusa. No, that's the Medusa-shaped one. Cestum was the really long one.
Okay.
It's the one that looks more like a Medusa in its shape.
This is dive 13.
Dive 13.
This is going to be water sample 2 when it's time.
13 D13O2W. And recall, we're going to be able to make 5 biological collections of specimen collections,
In the buckets.
or at least attempt 5.
Yeah.
But again, I think if our past experience holds, this one will also, although this at 500 meters will be easier to see, will still be a little more challenging than when we get another 200 meters deeper, farther from the light source above. And it's where everything seems to pop out.
Yeah. And we're going to be in the deep scattering layer here, so.
That's right. So hopefully we'll see high diversity of organisms. You can see D2 is dropped down, and yeah, we're right there.
Yep, we're at 505 right now.
This. What I want to see is a ctenophore.
Yeah. That narrows it down.
That looks like
that orange?
this one.
The orange one?
Olicatina. I don't think we will, but...
So we want to catch that one?
If we see anything like this, we want to catch it, yeah.
Okay.
It'll basically... I'll jump out of my seat.
Yeah. Okay.
And another one that would be really fun to see is Lampea. But I also don't know what our chances are. That's a specialist on salps.
Okay.
Here. And
I've seen those yellow ones, I think. The yellow clear one.
a little Lampocteus.
Mm-hmm.
They call that the blood belly.
Yeah, I can see why.
Yep. And we'll be doing our second water sample soon.
Mm-hmm.
We can definitely see. I think there goes a salp. That was a salp in the top left. Who knows? Maybe we will see Lampea.
Fish, 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 fish.
Yes. So things are already coming to the fore.
Mm-hmm. All right, I think we're about to take water sample number 2.
And here we are in the deep scattering layer. It's a nice siphonophore Fisonect up there.
Yep, go ahead. Thank you.
Fantastic. So now we have our second water sample for our environmental DNA collections. And we are in the deep scattering layer and ready to begin our second transect, about 500 meters of depth. Woohoo, nice big foram there. I think we're going to see a lot of forams at this depth.
Okay, watch leads and shoreside. This is the official start of the 500-meter transect. I have it at 1558 UTC.
Thank you. A little copepod or something at noon.
Pink Cooper, copy.
They're hard to catch.
All right.
Oh.
There's a Medusa, maybe?
Oh, it definitely looks like a Medusa.
Yeah. He's got really long tentacles.
He does. Nice and
Copy.
They're curled up.
white-tipped. Can't really see the number of radial canals,
I think.
but it has tentacles pointed forward and dangling down. Or is that? It's not 2, is it?
2 radial canals? I don't know.
No. I meant 2 It's not too Medusa or
two
anything.
critters.
No, it's not.
He's got a lot of tentacles.
He does. Just can't quite make it out. But that was cool. I'll have a go at that video later.
I think Hydra Medusa, something?
Definitely a Hydra Medusa. Likely a Trecaline. Oh, almost certainly a Trecaline. So we got a little fish coming in here.
Is it bristlemouth, maybe? It's very shiny.
It is very shiny. I think this is more like
You can see his dorsal and anal fin are pretty tall.
you can see its eye fairly prominently.
Big mouth.
This is a lanternfish.
Maxine.
Great footage of it.
That's teeny tiny, that little guy. He's probably like a centimeter.
Definitely not cyclothony, so I think it might have been a kind of lanternfish, but we will get our nice report from Ken Sulak or someone else
Yeah.
who are good with the fish with all the fish captures.
And a fishy goes by.
Mm-hmm.
Okay.
Something white. Ooh!
Something gone.
Nope. Oh,
It's one of those fish that we just saw, probably.
a little fishy.
Is it the same? Oh, no, I think that is a cyclothony, right?
Okay.
Bristlemouth.
Okay.
The most abundant vertebrate on the planet. Population estimated at over 250 individuals.
My goodness.
Quadrillion?
Quadrillion. So we did this math, or Matt did the math, and it was, if a million were seconds, it's 32 days?
Oh, you're going to ask
A billion, I may have to look that up again.
a billion. A quadrillion is a large number. It's 1,000 trillions.
Wow.
Yeah. Okay. A million seconds is 12 days. A billion seconds is 31 years. A trillion seconds is 31,688 years. And then a quadrillion is like 42 million years.
42 million years.
Yeah, something like that.
Quadrillion seconds. Yes. So there are a lot of those fish.
White dot.
Look how this is moving. Oh, yeah, let's get a look at this. Look at that. It looks like
Cool.
You can take more if you got it. Oh, it's Max. He's a ways away. Happy?
So the first fish we zoomed in, Mike Vecchione says, might have been called Bonapartia. I'll have to... Oh, now we get a good head-on view of this guy.
Ooh,
Okay, how about this side
that
view?
looks similar too. He's got the same dorsal and anal fin.
Uh-huh.
That's the last one. There's a copepod up there.
Hey, Okeanos, Mike Vecchione here.
Hey, Mike.
How are you?
Hey, I think that was another Bonapartia.
Bonapartia?
I'm okay. I'm enjoying the dive. But again, I'm not a fish person. I think that that really long anal fin is characteristic.
What's this white dot?
Kind of a bristlemouth fish.
Mm-hmm.
Oh, okay. Different kind of bristlemouth.
Oh, yeah, there's a lot of different genera and species within the bristlemouths.
Thanks, Mike. Got something swimming.
So I'm going to hang up and not embarrass myself anymore.
Oh, please. I know nothing, so, you're doing way better than me. There's something moving. That's about all I can give you. Ctenophore. Schmutz, ctenophores, and fish. That's the extent of my taxonomic knowledge.
Nice little crustacean zipping by there. It's a small amphipods or something like that, swimming through.
There's something moving up here, yeah? Little siphonophore?
Most definitely a siphonophore.
Bloop, bloop,
Take it.
bloop.
Yeah,
There you go.
that's nice.
It's got like
It's a
6
fisonect.
bells.
Yeah. That could have been another of the nanomias.
Okay. I guess another fish. Oh, actually, no. What is that? Garbage? Okay.
That wasn't nothing.
There's something. Probably a foram.
Mm-hmm.
This one looks like a tiny spoon.
It's down here, Ron.
Ooh. Schmutz.
Yeah.
I think the most abundant stuff out here is schmutz.
It's all this indication of other life and feeding and pooping going on.
Yeah.
And shedding of mucus that entrains these little particles in the water.
Mm-hmm.
Makes for good detritus food at the bottom.
Mm-hmm. Also feeds the Lophelia reefs.
Yes, exactly. They need that input from above, don't they?
There's a siphonophore at 3.
Just to the right.
What's this, Ron?
Little white thing. I don't know.
Something looks similar. We zoomed in on at one point, and then it swam away really fast.
Maybe a fish.
So reflective.
Maybe
It's hard to characterize.
maybe a mollusk. Oh, I think it's a fish
I think
pointed up. I think he's looking up.
Mm-hmm. See what looks like the head end.
What was that?
Another one.
Oh,
That's got that cyclothony swim look to it, right?
Mm-hmm. There's another fishy. He's pretty clear. There's a siphonophore at noon.
Straight ahead, yeah. Looks like a prayed. You can sort of see the front end there reflecting at the big beats of its swimming bells. I think that looks similar to the one that we were seeing a lot at the 300-meter transect.
Thanks.
What is going on here?
Ooh, is that ink?
Yeah, I think so. Looks like squid ink to me. And then there's something behind it, a little ring of something.
Huh.
We just missed the squid.
Dar.
There's something at 4. Fish, maybe? I think so.
It's very energetic.
He looks very excited.
Mike Vecchione in the chat, he agreed with our grr. I think it would be great to actually collect a squid
Oh, yeah.
for Mike. Would have to be something small and not strong swimming. It's probably rules out a lot of them.
Yeah.
Yep.
Cadignath or a fish? One of those fish looks like a pin.
Minimum focus.
I
Really
think so.
tiny, tiny?
Yeah. I think we saw something like this on the first.
Huh.
Really hard to make out.
Mm-hmm.
Let it go. Keep moving forward. Thanks.
What are you seeing?
Yeah.
There's a white thing at noon.
Go for white dot.
We've got
Foraminifera.
foram. Yeah.
The fuzzy foram.
Really intriguing looking, these forams, I think.
Little starburst.
Yeah.
Looks like they have glass almost.
Need to hang it on the Christmas tree.
Mm-hmm.
Coming right to left. it's a fish, right?
I think it's a fish. It's a very tiny fish. I think it's the same one with that long, right? The long dorsal and anal fin we saw.
It's the maybe Bonapartia. On there, we got a nice look at the top of it too. That's good for more.
Cool.
Oh,
nice.
Really nice view.
Big black eye.
Right.
So this thing's less than a centimeter, I think. It's tiny.
Super.
Yeah.
So much bigger eyes than cyclothone.
Thanks. Yeah. Sometimes it works. Yeah.
There's a shrimp or something going off screen. I may have been a Cadignath.
Saw one of those X chromosome shaped Phaedarians flying by on the left.
3 dots.
Oh, that's neat.
Pieces of that same siphonophore, you think?
I think
Yeah.
so. Looks similar,
Take more.
but crunched up. So it's all
Yeah.
squished together.
Oh, yeah, look at that.
Oh, yeah. That's the front end of that parade.
Let this
Oh,
one-
yeah, you can see this really nicely.
Kind of a small one. I keep going.
I think
Yeah.
that's
Cool.
the definitive view for someone to be sure we got the name right.
It looks like he's got lots of bells. So each one of those little circular things on his
Also
on
has
his
a-
tail it looks like it's got its own bell.
Yeah, some of them release what's called this eudoxid stage, like a little mini colony.
Do they break off and then form their own family? Yeah. Looks like a pair of antlers. 6 o'clock.
Yeah, I saw that too.
Antler-shaped splits. Medusa-shaped splits.
Yeah.
String of dots, siphonophore-shaped splits. Got
Tiny
another
fish.
Tiny fish. Now this is giving me cyclothone vibes.
Yeah. Dorsal and anal fin are the same size, but really big. There you go.
Yep.
Nice and
And
clear.
kind of far back on the body. I think we got cyclothone for sure.
Go out video. Thanks.
I feel like if I did maybe 100 of these expeditions, I'd get pretty good at this.
Yeah. There's something, Medusa or... Yeah.
Oh, definitely a Medusa.
Really
Great if we
long
can get-
tentacles.
Yeah.
Cool.
All right.
So it looks like tentacles are above the
Above
bell?
the bell,
right?
yeah. So it's another kind of Narcomedusa. And then something, if it doesn't have stomach pouches, we would probably call this Solmaris.
Okay.
But we can't really quite see well enough to know if there are stomach pouches or not. But I really like this.
Twenty-five tentacles, I think. I don't know if that matters, but
It's one of the things we definitely count when we're figuring out what's what. Well, thanks for that. But yeah, I think that's Narcomedusa and most likely a species of Solmaris for